Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video, we will be learning about the DABT topic, statistics, P-value, NOAEL. Let's directly go to the question. The first question is, in evaluating differences between mean value of clinical chemistry variables from an animal toxicity study in on day 1 versus day 14, statistical analysis are used and a p-value of 0.05 for several comparisons were divided. What does a p-value of less than 0.05 means? See, important thing you have to understand from this question is one question on statistics you will be getting. Okay, and here the statistics where you will be using is whenever you are performing any toxicology study. Okay, then you have to decide p level of significance. If you look into p level of significance, here in this question it is given as less than 0.05, but you will be also having when you are performing research it will be p less than 0.001. Okay, very strong evidence against the null hypothesis. Okay, uh, let's do one thing. Let's go through the question, then I will be explaining about the p level of significance. I will be explaining. I will also explain about the null hypothesis h0 as well as h1, that is how to reject the null hypothesis h0 and h1. I guess h0, let's see the options, then we'll understand. The option is the difference between the variable means are statistically and biologically significant. The significant difference between means are caused by the treatment. The probability that the observed response had no effect is less than 1 in 20. The probability that the observed response had effect is less than 1 in 20. Okay, so what is important is we need to understand what does the p value 0.05 will mean in this particular case. Here the right answer is, as you can see in green text, is option C. What is option C? Option C, the probability that the observed response had no effect in less than 1 in 20. Okay, out of 20, 1 in 20 it did not have any no response. So let's see what is the explanation and difference which is given by American Board of Toxicology. As I said, no, these questions are taken from this particular 50 questions quiz which was uploaded earlier in DABT site. Now it is no longer available. So so we'll see what is the explanation which is given by this DABT is the significance levels is the chance of obtaining a false positive result due to sampling error. So whenever this is known as type 1 error. Okay, so whenever you are performing clinical chemistry, here the clinical chemistry is biochemistry, whether AST, ALT, GPT or glucose levels, those things based on whether you are doing liver parameters or kidney parameters, various clinical chemistry essays will be there. Okay, in that you will be getting some readings. Some readings will be outliers. Okay, some readings will not be in line with the readings. For example, you have taken n equal to 3 observation, 3 readings. One it is giving 50, another it is giving 40, another it is giving 10. Or if you have 4 or 5 samplings more, another it is also giving 45, or another it is giving around 51. So the odd number, whichever was the 10 or 20 is, that has to be removed. That is called removing. So certain times also happens when you are doing biochemistry parameters note, you have to pipette in your append of so and so volume, like around if it is a zero point 0.5 ml or 1 ml, like that, when you're putting the sample in the centrifuge. But if in case you have not done the pipetting properly, okay, complete the volume that is needed for the sample to be analyzed in the centrifuge is not transferred into the append of, which will be kept under centrifuge, then you will be having error will be having that particular. When you put that sample under reading, you will get false reading. So that is called sampling error. Okay, so this is nothing but type 1 error. So we have in statistics, you have type 1 error will be there, type 2 error will be there. So go back and try to read what is type 1 error, what is type 2 error, what are the examples which comes under type 1 error, what are the examples which comes under type 2 error. In this case, the level of significance p equal to 0 less than 0.005. In this case, this is something called sampling error. Once again we'll read, because it might be new thing to certain students who are appearing for the exam. So the significance level is the chance of obtaining a false positive. Okay, actually it has to give negative result, but it is giving a positive result, and that too it is a false positive result due to sampling error, known as type 1 error. Okay, it is usually set at 5%, although lower levels are sometimes specified. Here lower levels के लिए first example is nothing but p less than 0.001. In this case, what is a 99% confidence interval you are taking. Here in the case of here 95, 99. In certain cases, you will take 99.99 percentage of, so you are not allowing, we are allowing only 0.001, only you are allowing. If you are taking 100 samples, that much only error you are allowing in your null hypothesis, in your research results, when you are analyzing the results. Okay, particularly if you see the statistics now, for social type of experiments. Okay, when you're collecting epidemiological data, for example effect of internet usage on the student, something like that, no, social collection of the data will be there, no, there you can use up to 95% confidence interval. But coming to health sciences, you do not want even a single patient to die. Okay, so in such cases, in human clinical trials, we use p level of significance to the highest maximum, maybe 99% or 99.99%. But there is an exception in the case of COVID trials, the clinical trials which were done on COVID vaccines. There, the p level of significance was little bit lesser, not 99.9 percentage. You can go back and check what I'm trying to say, because in such an emergency case, no, if we stick to the stringent time lines like p level up to 99.9% means the success of the drug to achieve 99.9% significance will be difficult, and the vaccine or whatever was is difficult to come into practice. So they allowed that exception in that case, but not now, not all cases. So you have to understand social studies, whatever social type of research or data collection is there, there you are using the p is p level 95% confidence interval also. Here in animal trials also we are using p level 0.05. If you see GLP studies, we will be doing like that only. And coming to clinical trials, when you are doing human volunteer studies, when you are doing, at that time you have to go for higher confidence interval. P level should be either 95%, 99% or 99.9%. Okay, so it is used at set at 5%, although lower levels are sometimes specified. In other words, it is a probability that the null hypothesis is true. As I said, no, H naught. So before initiating any research, we will frame a hypothesis, and always your hypothesis should be null hypothesis, opposite to the objective what you are trying to achieve. That is what you have to put in your null hypothesis. Then you have to reject your null hypothesis. You have to go opposite to what your objective is. Okay, so those who are doing research, they will understand what I'm trying to say. Statistical significance tells the investigator nothing about the biological significance, right? Statistical significance, it is a number actually. Okay, if n value for n observations are 3 or 5, and all the 5 are showing positive results, but that result might not have any impact or any usage in the biological inference. For example, how I should say, for example, people eating pizza in Hyderabad and versus the effect of rainfall in Hyderabad. So people eat pizza or not eat pizza. If you collected data, if you put it in your, there are a lot of statistical softwares are available. If you're taking these two different observations and try to put it, still it should show some kind of level of significance. Statistically, there is significant will be there based on number of samples. Like you are taking 100 people in Hyderabad who are eating pizza and effect of the rain on 100 on that particular day or a certain period of time. Still, you will find some data, some inference you will get it when you put it in the software. But logically there is no connection. That's what we're saying. There is no biological significance will not be there, even though statistically significant होने के बावजूद it will still not have any biological significance. For example, in the case of animal trials, maybe there is a change in the body weight or something like that. Statistically there might be, or urine color might be changed, but that urine color change, if it, even though it has a statistical significance, it might or not have any biological significance. Getting my point? With the treatment of the drug, something like that. Okay, So once you start analyzing the data, you will understand what I'm trying to say. When you are doing an experiment, you will start getting the results. So you have to correlate that results to the test item, what you are administered, or to the chemical in which the chemical which you have administered to the animals, right? Yeah, there should be a correlation should be there. If there is no correlation, still statistical significance is there, then you can ignore that, but you have to capture in your final results. Okay, so let's see once again last statement. Statistical significance tells the investigator nothing about the biological significance or whether the difference are true or not. So please go back and read Hayes 6th chapter page number 377 in detail. So from this particular question, what you have to learn is about the p level of significance. You learn null hypothesis, you learn, and you please go back and read type 1 and type 2 error. In another video I'll definitely bring the comparison between type 1 and type 2 error with examples, and also important biological significance and statistical significance. One question on this you will definitely get. And you, those people who are doing PhD, or those people who are doing research, who are analyzing the data, or there are some biostatisticians who are preparing for the ABT exam, they will definitely know what we are talking about. P less than 0.01 or 0.05. Okay, p level, this table also you try to remember. Okay, let's directly go to next question. Okay, the next question is something related to NOAEL. Let's see what question we have. The following dosages of a test drug were administered to male rats, okay, by oral gavage daily for 28 days. So this is a 28-day repeat dose toxicology studies, that to male rats, not female rats. So the doses were 10, 50 and 100. The only observed toxicity was focal hepatocyte necrosis. So remember, this is something related to liver cells. Okay, observed at 100. So at a high dose, 100 mg per kg, there is an adverse effect on the liver. Okay, liver cells are dying. Necrosis is not even apoptosis. Which is a irreversible damage. Based on these data, which is the what is the NOAEL. So like this, you will get in your exam. They will give you a case study, they will give you the doses, and they will ask you to derive NOAEL based on the data. So let's see what options they are giving. Option A is 10 mg per kg. Option B is greater than 50 mg per kg but less than 100 mg per kg. Option C is 100 mg per kg, and option D is 50 mg per kg. So the right answer, as you can see in the green text, option D, which is 50 mg per kg. So let's see what is the explanation and difference which is given by American Board of Toxicology. The NOAEL is the professional opinion. So those persons who are working, the principal investigator or study director, is a professional opinion based on the design of the study. In this case, the design is low dose, mid dose and high dose. Indication of the drug, expected pharmacology and spectrum of off-target effects. Okay, if our chemical whichever we are administering to the test system animal, we want the drug to go and act on certain like if it is a brain targeting or it is a liver, but it is going and affecting heart. So off targeting effects, on targeting, off targeted effects, that you have to remember this terminology: on target, off target. Okay, it requires a decision of what findings are adverse to the test system. Your test system is male rats, under the conditions of the study. Okay. Then next is, so this is the guideline which we are recommending, which was recommended by ABT. Go back and read this website. You can go to FDA website. You can see CEDAR will be there, CBAR will be there, CBAR, biotechnology did there. So in that they have guidance document is there. This particular guideline I have already mentioned in my previous videos also. This is the guideline for the industry estimating the maximum safe at starting dose in initial clinical trials for therapeutics in adult healthy volunteers. So before initiating phase 1 clinical trial, this particular guideline will tell us how to decide the first in human dose. Okay, this is the maximum estimating maximum safe starting dose, FIH, first in human dose. So you will take an animal study, you will get a NOAEL. That NOAEL fractionation you will take, and you will derive HED, human equivalent dose. That HED will be your starting dose for your phase 1 phase 1 trial. Human volunteers of first dose. Okay, so please go back and read this particular guideline in detail, because in this guideline, in my previous videos I have kept the screenshots also. There will be based on your Km constant, body surface area. There are fixed correction factors are there. For rat you have to use 5, or for mice you have to use 12, or some numbers are there, 6.1, 3.2. For rabbit, like that is there, where you will take animal NOAEL, convert it into human NOEL, or that will be considered as your HED. Okay, so please go back and read. So you can also refer to Cassett and Doll 8th revision, page number 134. Okay, and 9th also they have given the pages 36 and 37. So please go back and read. So again, remember the important thing that we have to learn from this particular slide is that you will get such case studies. Those who are working in GLP labs, those who are working on repeat dose toxicology, 28-day repeat dose toxicology, study directors, study personnel, they will easily know this question. They will be easily able to answer this question. So they will be giving you such cases will be there where they will give you the scenario of the study and the doses, and they will ask you to derive NOAEL. Okay, this is based on the observed effects which are given in the question. You have to derive a NOAEL. Okay, so you can see this is a typical dose response curve. S-shaped curve will be there. The NOAEL is the dose at which no adverse effects is there, and LOAEL is the dose at which lowest dose at which adverse effects are observed. So in this case 100 was the dose at which liver adverse effects on liver were observed. 10 and 50 were the doses at which no adverse effects were observed. But out of this also, you have to consider, as per the definition of NOAEL, you have to consider the highest dose. So out of 10 and 50, which is the highest dose? 50 is the highest dose at which adverse effects are observed. That's why 50 is your NOAEL. Okay, and in this case, LOAEL will be, for example, 100 is there, and there is another dose like 200 is there, another dose is there 500. Like for example, in this study 5 doses were there: 10, 50, 100, 200 and 500. 100 you have some saw some adverse effects on liver. Okay, and at 200 also you find 500. 500 so you have completely seen that liver failure is there. At 500 person has died, but the test system animal has died because of the liver toxicity. So all these three doses, 100, 200 and 500, any dose above 100 is it is causing toxicity. Okay, adverse effects out of three doses, which is the lowest dose at which adverse effects are after 100, 200 and 500? 100 is the lowest dose, so this will become LOAEL in this case, in that case. Okay, so yeah, to split the study, try to understand this terminology. This is a typical S-shaped curve, dose response curve, in which x-axis dose will be there, response will be on the y-axis. As you people learn in detail more about dose response curve, there will not be only S-shaped dose response curve, there will be U-shaped dose response curve, J-shaped curve will be there, U-shaped inverted dose response curve will be there, hormesis curve will be there. So there is a separate topic you can learn about the dose response curve. Okay, so this dose response curve again, it is based on the basic principle which is suggested by the Paracelsus, that dose will determine the substance is poison or not. Okay, so anyways, let's finish this. Uh, Thank you so much. Be curious. For any questions, please write mail to so and so mail email ID. Like, subscribe, and follow my YouTube channel Toxicology on Board. Thank you. All the best for your exam.